Greetings, everyone. Welcome to my sessions. These sessions are basically directed towards having a basic idea about immunology, how things work in our immune system. I tried to give you an overview of the different components of the immune system here, and over the course of time we'll have elaborate discussions on cells and the mechanism of how the system works. But today in this particular session I want to give you a brief overview of the lymphatic system. Now all the material that I'm going to cite are freely available. I'll be discussing material from papers which are freely available. I'll be talking, my course of lectures will be basically the series of chapters that you get in Kuby. I'm using the 7th edition, so if you have the book, you can just follow what I'm trying to deliver here. But the materials I'll be discussing will also include a number of free papers which are available on the internet. I'll be using internet sources, so all the material that I discuss will be at your disposal. I'll also try to share the links so that you don't have any problem. Why I am starting with the primary, as you can see here also, I have highlighted the primary lymphoid organs and the secondary lymphoid organs. Why I want to begin here, because the two most important, whenever a pathogen enters your body, it has to be detected, and your body immune system has to initially know about that there has been an invasion. See, first what I like to deliver here is a pathogen is something foreign to you, but if you look from the angle of a pathogen, it wants to survive in you. So there is a competition between your body to identify the pathogen, and on the same side, there's all the attempts are being made by the pathogen, the so-called pathogen, to survive in your body, because it is the survival of the fittest after all. So you don't have to just think about how your system is developing mechanisms to delete or to get itself free of that pathogenic load, but you also have to think about, in terms of a pathogen, how are you trying to evade the system. So my lectures will aim at providing you these ideas. All right, why am I beginning with the lymphoid system? Because whenever you talk about the B cells, the T cells, they are all derived from the lymphoid system. We are going to talk about hematopoiesis, and we are going to talk about myeloid progenitor cells and lymphoid progenitor cells later. But what is lymph actually, and what is the lymphoid system? So as you can see right now, what we are discussing here is from Kuby. This is the 7th edition, and this is the chapter 2 on cells, organs, and microenvironment in the immune system. And the first thing they have highlighted is the primary lymphoid organs and the secondary lymphoid organs. Now my suggestion to you is always try to understand each and every concept as you proceed. Now we try to mug up a particular concept and then this basically distorts the entire process. So if I may make a suggestion here, try to understand each concept and then move ahead. So I'll try to deliver, at least give you an idea about every term that we are discussing. All right, and then after understanding that, only we move ahead. So the first two words that we get here, when we look into Kuby, is the primary lymphoid organ and the secondary lymphoid organ. So we have to have an understanding about what is primary lymphoid organ and what is secondary lymphoid organ. See, the genesis of all the cells in your immune system happens from a stem cell. We all know what's a stem cell. It is a cell which has the capacity to become any type of cell. So genesis definitely has to be from a stem cell, but stem cells also need an environment to grow. If I want to become a good teacher, I need a proper environment. If you want to study, you also need a proper environment. So these environments are offered by definite institutions, by definite locations within the body where cells grow and when they mature, and finally you are supposed to teach. One of such locations that we have is the primary lymphoid organ. Here the genesis takes place. Here all the cells that we'll be talking about are formed. But then again, we have the secondary lymphoids, where all the other processes, where you basically expose your cells to the foreign antigens. Now we'll be talking more about it. Whenever we talk about the primary lymphoid organs, it's basically your bone marrow and your thymus. Now, you all know, I believe that you understand that hematopoiesis takes place in the bone marrow, but the T cell development and maturation will be taking place in the thymus. Now, I wanted to show you, so bone marrow, everybody knows what a bone marrow is, where it is located. My bigger bones, my big bones, my ribs are the places where the hematopoiesis is currently happening. But what about the thymus? Most of us, we do not know where the thymus is, and so I was interested to show you how the thymus basically looks like. I'm lost here. All right, so this is the thymus. Now, as you can see, this is the website from where I'm getting my materials, or you can refer it, and it is a free to access website. So right above the heart, as we can see here, I have my thymus. It is the left lung, and right above the heart I have the thymus. So now you know it. So this is and you are supposed to keep it in your head, so that you know when you conceptualize, you see the thymus where it is. So this is the thymus right above the heart, below the left lung. As you can see, below the lungs, above the heart, we have the thymus. Fantastic. So what are the secondary lymphoids? I'm not going to focus today. My lecture will not be focusing on bone marrow and thymus, because about bone marrow I'll be discussing when I'm going to talk about the B cell development and the maturation process. About thymus I'll be talking about when I'll be talking about T cell development and the maturation process. So my focus today would be trying to deliver to you what is lymph, how does lymph originate, and what are the secondary lymphoid organs. Now, again, I'll be referring to one of these papers here. This is an old paper. As you can see, this is a 2009 paper, but the language is fantastic. So I guess, you know, this is good enough to give you an initial idea. We'll be also going through a number of other papers. This paper here, this is a recent paper comparatively. This is a 2016 paper. Now, why do you need secondary lymphoid organs, and what is lymph at the first case? See, when we are pumping the blood through the vessels, so when we pump blood through the vessels, we know there are veins and we know there are arteries, and these blood vessels are lined by endothelial cells. Now, you need to know about endothelial cells. Please Google it up. Please learn about what are endothelial cells. And these endothelial cells line the vessels. All right. Now, just imagine you are pumping water through a pipe with high pressure, and if there's a small hole in that pipe, the water will try to seep through and it will just form like a fountain, right? So same when the heart is pumping blood, when you're forcing blood through all these arteries, these veins, and all the related structures, the blood, which is basically made up of cellular components and the liquid component being the plasma tends to seep through these endothelial cells which are lining the capillaries. All right, I'm just trying to give you a very basic understanding here. So these endothelial cells which are lining the capillaries, which are lining the vessels, through them the fluid component, that is the plasma, including certain proteins, they tend to seep through. And why did nature allow this lymph to seep, this plasma to seep through? We are still not going to call it lymph. This plasma, it goes and washes away all the small factories which are operational. Now, you have to think about your body as a factory. You have so many cells, and all the cells are doing all the metabolic activities that sums up together for life. Right? We are alive because of those cells doing their metabolic activity. But then whenever there is metabolism, there is always also production of waste. How do you wash away that waste? See, all these days, you know that blood is responsible for helping in the gases exchange, for providing nutrition to the cells. But what takes away the waste? So this is nature's way of washing the cells. You know, I wash my cells. So the plasma seeping through the gaps in the endothelial cells will be going into the tissue spaces, will be washing the cells. But then after you are done washing the cells, this fluid has to be collected back. Otherwise, you will suffer for something which is called edema, a swelling where the fluid is accumulating. So you need to then develop a system of vessels which is supposed to collect all the plasma that you have, that seep through your vessels, wash these cells, and this plasma, after it has gone into the tissue spaces, we now call it lymph, and this lymph has to be collected by vessels, and this is the lymphatic network. So I believe now you have an idea about the lymphatic network. You see, The lymphatic network is basically made up of lymphatic vessels, lymph node, lymph follicles, and then one of the most important components is the secondary lymphoid organ. So we'll be talking about more about it in time to come. So now you have a preliminary idea about lymph. Apart from washing, so see, say you have a house here, okay, and there's another house there in your neighborhood, and there is this person who collects the waste from house A and house B, right? Just by looking at your waste profile, just by looking at the waste profile of house A and comparing it with the waste profile of house B, I can have an idea about what are the food preferences of this house, right? So similarly, when the lymph is washing away the cells, the lymph is not only washing away, taking away the waste, but it is also taking away cellular debris. And this cellular debris also includes pathogens, pathogenic debris, which can be derived from a pathogen which is residing inside the cell or which is residing outside the cell. Right? You getting my point? So a pathogen, see, I told you we have to think about immunology from the perspective of even the pathogen. Now the pathogen knows that, you know, if I stay outside, if I stay in the circulation, the chance that I get detected is far more than if I try to hide inside the cell. So parasites, pathogens, try to hide inside the cell as well. But when this pathogen is replicating inside the cell or doing its own metabolic activity, it also generates waste. So the lymph, which goes and washes away all these cells, is not only kind of collecting waste from cells, from a healthy cell otherwise, it also collects debris and waste from pathogens who might be residing inside the cell or pathogens who might be residing outside the cell. And when you collect this lymph and you analyze it at designated places like our secondary lymphoid organs, you get an idea about if there is a foreign antigen, and we'll be talking more about it in the time to come. So what does a secondary lymphoid organ do? As in this particular paper, look at this. Given the great diversity of somatically generated BCRs and TCRs, these are B cell receptors and T cell receptors in each individual, there's a requisite low precursor frequency of lymphocytes relative to a given antigenic epitope. Now, whenever we talk about antigens, antigens have, any pathogen will have definite antigens, and antigens have epitopes. You know, these are epitopes are things which are under, which are identified by paratopes or the antigen binding sites. So B cells, which are responsible for the humoral immunity, will have BCRs, that are B cell receptors, right? Similarly, there are their relatives, that are the T cells, will have TCRs. Now B cell is responsible for humoral immunity, whether T cells, we will talk about T helper cells and T cytotoxic cells, T regulatory cells, so many of them. But T cytotoxic cells are responsible for the cell-mediated immunity. Now we'll talk about it later. But just imagine your body has the capacity to identify millions of different type of antigens. This is only possible because your body generates so many different type of BCRs and so many different type of TCRs, right? But each cell, a distinct B cell, a small B cell with a distinct BCR, will not amplify until it is stimulated. So each distinct B cell with a distinct BCR, or each distinct T cell with a distinct TCR, will be there in your circulation. That can be blood, that can be your lymph, but it is only available in few copies. So just think about that. A particular B cell with a particular BCR is here in this part of my body right now, and the pathogen has invaded through my limbs. So there is a very less probability that this particular B cell will circulate and then it will identify that particular antigen and then it will be stimulated. So you need places where you bring your B cells, where you bring your T cells in close proximity with these antigens, and the secondary lymphoid organs are doing that particular function. So thus there's a requirement for physical structure or location in which both the antigen restricted lymphocyte would mean that a lymphocyte which will only respond to a particular antigen because it has, after all, got released into the circulation after maturation, and a foreign antigen can meet without any dedicated tissue set. Without such a dedicated tissue site, the likelihood that the antigen and one of the few potential reactive lymphocytes encountering each other within a vertebrate's body is extremely low. Secondary lymphoid organs emerge in evolution to provide a structural framework necessary for the concentration of antigens and antigen-specific lymphocytes to initiate an effective adaptive immune response. So I believe you understand that. So they are not just responsible for accumulating the antigen, they are also responsible for, inviting the lymphocytes to that particular position. So if I'm a secondary lymphoid organ, look at this. If I'm a secondary lymphoid organ, my responsibility is not just to, collect all the antigen. How am I collecting all the antigens? Because, the lymph which is washing away all the cells is getting collected at the secondary lymphoid organs. All right. So my responsibility is not just to collect the antigen and present the antigen. My intentions are also to invite, to attract all the lymphoid cells. That can be B cell, that can be T cells, so that these specific antigen restricted, see the word, the antigen restricted lymphocyte comes in close proximity with the specific antigen, and then the process of adaptive immunity is triggered. All right. So they must also possess mechanisms for the recruitment of lymphocytes and for the acquisition of and retention of antigen. Acquisition is how the lymph is bringing the debris into your secondary lymphoid antigen, into their secondary lymphoid organ. That is acquisition. But what do you understand by retention? Retention is this antigen. See, this antigen is in a fluid, right? So it can be lost, but you don't want to lose it, so it has to be retained. How do you retain it? We'll talk about some antigen presenting cells in some time to come. All right. So now we go to the other article, and I'll discuss it as we go through. All right. So during antigenic challenge, additional lymphoid tissue are apparent in the lungs. All right. So lymph nodes. So the first thing that we have to talk about here is different type of secondary lymphoid organs that we have. All right. So when we talk about the different types of secondary lymphoid organs, these include the spleen, this includes the lymph nodes, this includes the pear patch, this is there in the ileum, we have the tonsils, we have the adenoids. All right. So before I go into the structure, or before even I discuss about how things are working there, let's try to have a small understanding about the secondary lymphoid organs. We'll talk about the spleen later, we'll talk about the lymph node later, but I want to give you an idea about the tonsils and the adenoids. Now see, tonsils and adenoids, for that we'll take the help of certain websites. Okay, fantastic. So if you look at the tonsils, everybody has tonsils, I know we have tonsils, but where are the tonsils located? They are a set of masses which are located on the back of your tongue. So as you can see, we have the tongue, we have the pharynx, and on both sides we have the tonsils. All right. And the tonsils located rear throat of the pharynx. Each tonsil is composed of tissue similar to lymph nodes. It's covered by a pink mucosa, and it is responsible for screening the air that you are taking in. So just to give you a brief idea, okay, we understood tonsil. Let's understand adenoids, and I'll be talking about something. What are adenoids? The website that I'm referring to, it is freely accessible and you can directly see it here. What are adenoids? Adenoids are patches of tissues that are high up in the throat, just behind the nose. So if I'm talking about respiration right now, see, I don't want you to mug up the concepts. Try to visualize how the body is protecting you from foreign invasion. So if I'm breathing, the air goes in, and then there are the adenoids. Where are the adenoids? Just behind the nose. And after the adenoids, I have the tonsils. So why has nature set them up? They have been set up to capture or to identify any foreign particle which has entered. You see, very interestingly, all right, adenoids usually start to shrink after the age of about 5. So after after I have age 5 and then I approach towards my teenage, the adenoids will start shrinking, and I believe that at this particular point I may not be having the adenoids. So this is a fantastic concept where you say when I was a baby, when I was growing in my mother's womb, and then I was born, my immune system was basically handled by my liver. And then because hematopoiesis at the initial point of time, in the initial times, happens in the liver because the bone marrow in a kid has not formed yet. And similarly, when the kid is growing, you also have the adenoids, and which are lost as I become an adult. All right. So now we know that two secondary lymphoid organs which are there looking after the air that are taken are the adenoids and the tonsils. All right. Fantastic. So going back to our literature, now we need to talk about the secondary lymphoids, right? So how are the secondary lymphoids working? So for the secondary lymphoids to work, you need to have both a blood circulation to the lymphoid as well as the lymph circulation to the lymphoid, and that's why that's the lymph. During antigenic challenge, additional lymphoid tissues may appear. The blood vessels to secondary lymphoid organs allow antigen access. So it is not just that the secondary lymphoids are only being supplied with lymph. The secondary lymphoids are also supplied with blood vessels. So, the pathogen may be there in your blood, right? The pathogen may be there in your tissue. So you need to sense whether the pathogen is there in your blood or in your tissue. Forget about it. You need to know if there is a pathogen. And where can the pathogen be? It may be either in the tissues or it can be in your circulation, right? So that's why both of them need to be screened. So both the blood and the lymph are coming to your secondary lymphoids. All right, fantastic. Lymph nodes. So the lymph nodes are a kind of secondary lymphoid, as we all know. Lymph nodes, which also serve as lymphatic, which are also served by lymphatic vessels, are effective in mounting response to antigens that are present in the tissue. So a lymph node is a secondary lymphoid organ which is supplied both by blood vessels and by lymphatic vessels. All right. These antigens, please notice, these antigens originate from foreign invaders that are transported by APCs or are derived from self-antigens. I'll talk about in my sessions about APCs, but just for you to know right now, APCs are antigen-presenting cells. As I said, as we just discussed moments back, the secondary lymphoid organ or your secondary lymphoid organ is not just intended to retain. It is not just a place where the antigen comes. You are supposed to retain, right? As I said, the antigen comes into the secondary lymphoid, but you have to retain it. How do you retain it? You retain it with the help of APCs, as I said earlier. So APCs such as your macrophages, APCs such as your dendritic cells. We'll talk about follicular dendritic cells. It's a small difference. There's a lot of difference actually between follicular dendritic cells and dendritic cells. We'll talk about all of that. All right. Self-antigen, yes. If those who are wondering what is a self-antigen, self-antigens are antigens that are my own. The body needs to, your body, as your immune cells are growing, as your humoral immunity is developing, as your cell-mediated immune response is developing, you need to make those growing cells aware that, okay, this is my own and this is foreign. All right, this is very crucial, otherwise you suffer from autoimmunity, where your immune system attacks your own cells. Basically, it is identifying your own antigens as foreign and is mounting an attack. You don't want that to happen. That does not happen in everyone, because as these cells are growing, you tell them, you teach them that, look, these antigens are foreign and these antigens are my own. All right, but to teach them that these antigens are my own, I need to tell them, I need to show them these are my own antigens. So you need to also show them self-antigens. We'll talk about more about it in days to come. The capacity to discriminate between dangerous foreign antigens and benign self-antigens relies on APCs, their state of activation, the recognition capacity of naive T cells and B cells. Who are going to recognize? Definitely the lymphoid. So lymphoid are constituted by Bs and T cells. Dendritic cells constitute sample self-antigens and migrate to draining lymph nodes even in steady state. So this line is really crucial. You need to understand this. Even if you are not infected, even if you are not sick, These are basic processes that keep on happening all the time. Your lymph nodes will always bring in lymph and will always get an efferent blood supply, and these supplies will be screened all the time. And one such very nice story, look at this. This is just a story, right? Dendritic cells constitutively sample self-antigens and migrate to draining lymph nodes even under steady state. That's your basal state. Because most self-antigen bearing dendritic cells in the lymph nodes are immature, they do not effectively activate naive cells. So you do not have these dendritic cells. So just imagine, these dendritic cells which are not completely mature, they dig through all the matter that has entered the lymph node. And while they are digging through all the matter, they are also binding some of these antigens on their receptors. We'll talk about these receptors later. These dendritic cells, these APCs, are not good enough to activate a B cell, which can then stimulate antibody production. They are not good enough to trigger the activation of cell-mediated immune response. But yes, what they are good at is they regulate self-reactive T cells. So T cells which have matured in the thymus, they need to be screened whether they are reacting against self-antigen. So that test is done with the help of these dendritic cells which bear self-antigen. So if a particular T cytotoxic cell interacts with a dendritic cell bearing self-antigen, you basically trigger a phenomenon which is called energy. A-N-E-R-G-Y. What is energy? I would be in a state of suspension. Very basic idea. I will be in a state of suspension. I will not divide, and then I will trigger apoptosis and I will die. See, a cell which has responded to your own antigen, that cell should not amplify. Bad for your body. So that cell needs to be deleted. Nature does not give a second chance, right? If I have done a mistake, you probably would give me a second chance to rectify, right? But these are so pivotal steps, these are so important steps, that if a T cell is responding, is reacting to your self-antigen, that cell should not be allowed to replicate. So you impose energy. You are in a state of suspension, and after some time you trigger apoptosis, and then particular cell or that particular lineage of cells is deleted. Alright, so that is called clonal deletions. We will be talking about it later. And expanding regulatory T cells. Now, regulatory T cells are T cells which are responsible for regulating the T cell response, which also includes the T helper response, the T cytotoxic response, and we'll be talking about it later. Apart from now, as you have got some idea about the secondary lymphoids, what they do. So you see, I can tell you about this. The primary lymphoids are those, they are just like your educational institutes. They will give you, they will train you, but you do not have the basic understanding of how the industry works, right? You know what is good, you know what is bad, you have your degrees. So this is just parallel to your primary lymphoid organ, where the cells are developing and where the cells are amplifying, they mature. In the secondary lymphoid is just like your first job. It basically teaches you, okay, this is good, this is how you are supposed to react to this particular situation, this is how you're supposed to react to that particular situation, and if you're not able to react, you are deleted, right? So this is what happens in the secondary lymphoids. Apart from the primary lymphoids and the secondary lymphoids, we also have the tertiary lymphoids. Tertiary lymphoid organs, are more acutely tertiary lymphoid tissues, are accumulation of lymphoid cells that arise in an adult. Ectopic lymphoid accumulation. Ladies and gentlemen, ectopic means abrupt. These tertiary lymphoid organs will not be there all the time. Alright, these tertiary lymphoid organs are need based. They are kind of plastic. Plastic means their shape, their sizes will vary. So if there is an infection, if you have gone for an organ transplant, if there is an autoimmune response, you will get to see these tertiary lymphoid tissues. So they are aggregates of lymphoid cells coming together. All right. So I believe now you have an idea about the primary lymphoid organs. That includes your bone, that includes your thymus. Now we know a bone marrow looks like. Now we know what the thymus look like. We also know about the secondary lymphoid organs, which includes my spleen, which includes my lymph nodes, which includes my Peyer's patches, which includes actually what we missed is which also includes MALT, mucus associated lymphoid tissue, which also includes BALT, bronchus associated lymphoid tissue. Now I want you to understand what is mucous membrane and mucus associated lymphoid tissue, and for that, to understand that, you see, what is mucous membrane? Mucus, this is an article from Wikipedia, not a problem to get an access. Mucous membrane or mucosa is a membrane that lines various cavities of your body. So the moment I say mucus associated lymphoid tissue, the first word is mucus. What is mucus? Mucus, all we know is slime. So these cells will be producing some sort of slimy material. Why do you want you? Because you want to trap the parasites. Most importantly, look at the slide. Mucous membrane or mucosa is the membrane that lines various cavities of your body. So what are the cavities that you have in your body? You have the gastrointestinal tract, you have the respiratory tract, you have the reproductive tract, you have the urinary tract, right? So all these tracts will be lined by mucosa, right? And part of that mucosa will have some lymphoid tissue, which is known as the mucosa associated lymphoid tissue. Alright? Definitely you understand, they'll be releasing mucus, which will help you trap the pathogen. And then if you're trapping the pathogen, definitely the lymphoid cells there will be trying to retain that antigen and present it to elicit either a B cell or a T cell response. Fantastic. Now you know it. Alright. So next, what I want you to understand is how a secondary lymphoid or a lymph node basically looks like, and how those things work, how the secondary lymphoid organ tissues structure is, so that we get to know where the B cells really come, where the T cells really come, and we should also try to understand how the spleen really works, so that we get an idea how does these secondary lymphoid organs work. So in my next video, we'll try to discuss about different secondary lymphoids, so that we have an elaborate understanding regarding the same. Thank you so much.